So welcome back. This is lecture 6 in E443, 543. As always, I need to remind people that the class is live streamed and recorded and available on YouTube. I hope people occasionally, if they have questions, are looking back at those videos. We do try to go back and clean them up, although it takes some time to do that. If you find mistakes or suggestions for improving those online videos, those would be much appreciated. Pedro did a great job of introducing fields and diffusion last week. We're going to come back and go over a couple of details just to consolidate ideas, depending on how people feel in terms of their confidence. We can go through some of those things faster or slower. And then we're going to talk about secretion and absorption, which are the remaining really parts of the diffusion solvers that we're going to be playing with. So today, last time we talked about tracking fields, and Pedro did a beautiful simulation of contact-inhibited cell growth. We really skipped a little bit of the details on histograms, so I want to come back, and I hope people have that cell growth simulation from last week so that we can work from there. I want to talk about how to save plots and plot data. And then I'll show you quickly how to use text windows in Player. And then we'll very briefly review the diffusion concepts that Pedro covered last week. We may do a little exercise on initial and boundary conditions, although we can skip that if people feel that they're confident with it. And then I want to show you how to inquire values of chemical field so you can plot it in a scientific plot. And then we'll do that membrane exercise that Pedro started at the very end of class last week, didn't finish. And then we'll talk more about concept of diffusion length and diffusion time, which are very critical concepts in biology. They really govern a lot of the organization of biological structures. And then we will do some simple uptake and secretion exercises. So this is going to be more of a, Pedro did a lot of advanced simulation together. This will be more of a consolidation lecture. I hope it helps fill in some gaps. So the last time we talked about tracking fields. I think the tracking fields are actually one of the nicest things you can have that CompuCell has. Occasionally, the tracking fields will do some odd things, like if you have negative values, they may not display the way you expect them to. But the key thing to remember about a tracking field are two. One, that the tracking field goes in the init of your steppable. And two, that you have to create for each cell a dictionary entry that corresponds to the key that you're going to use in the tracking field definition. And once you've done that, it's all done automatically. The one thing that perhaps is a little bit unexpected is that if you have an intrinsic attribute of a cell, and maybe we should extend the tracking field definition so you don't have to do this, you have to copy that attribute into a dictionary entry. So if you want to plot a tracking field for the pressure, you have to copy cell.pressure, which exists as an intrinsic attribute of the cell, into something called cell.dict, square bracket, quote, pressure, close square bracket, so that it can be tracked. So you have to, that's the one manual step that you need to do. And so we did some examples of that last time. So last time we did a exercise with Pedro where you did cell growth in a tube and then put in a contact inhibition term for proliferation. And I'd like people, if they can, to bring that code up and run it, and maybe give me a thumbs up when you have that reloaded from last week. because I'd like to use that as the basis for what we're going to do now, going forward. Alex says he has it running. I'll give people a minute. If anybody needs help with that, we more or less have to go to Pedro for help with that one. Since that was a very elegant exercise, but not one that I had developed. He replaced the exercise that I had planned for that with this one, which I think actually is more fun and perhaps more informative as well. And I should also say that it's not absolutely essential that you have a specific version of that. We're going to be using that just as a demo so that we can do a little bit of exploration of how histograms work. I want to come back and remind you about the syntax of histograms. So, Emma, you've got it. Kyle, were you able to get that working?
I'm redoing all my install right now, so.
Well, we still haven't got reinstalled.
Well, I trashed my Apple version, and then I got a new PC, so I started from scratch and it's working now.
And
The true operational system now.
That,
you have Linux now?
I can.
But it's working.
Okay, what about people online? Alex, you said you had it. William?
Much further this time.
William, do you have it working? William, Jonathan, Santu? Can you give me a response if you got it or not?
I had to start over.
I had to reformat my computer over the weekend, so I lost a lot of my local files, which included this project.
Okay.
So
So
I'll just try to follow along, I guess. Or
no, so Pedro, could you please email everybody a zip of that exercise from last week? Again, the goal here was not to spend a lot of time redeveloping that particular model. We're not going to be using it except to explore graphics.
Yeah, I'll take 5 minutes to find it because it's been a while.
all right, thank you very much.
Anybody else? William, Jonathan, what about you? William, you say you've got it. Well, the other thing is if you have it, if one of you has it, perhaps you could email it to Santa or put it in the chat. Well, it's not so easy to put it in the chat because it's not a monolithic file, but if you can zip it. Do people remember how to zip a file? In Twit there's an option called Zip It, which will save a single zipped folder with all of the files needed, and Twit will automatically unpack those when you open it, so it knows how to zip and unzip the file structure. I'll give people a minute to get that. That was quite a rich simulation that Pedro suggested in that exercise, and one can do a lot exploring how contact inhibition works. When you have growth only, or whether you have growth under certain conditions and shrinkage under others, you can actually get quite an interesting variety of behaviors. So that's quite a powerful simulation. Sorry, was Santu, did somebody send that to you? Oh, yeah. Okay. So let me just talk you through histograms. The code, the basic histogram code, I know was given to you last week by Pedro. It's not that complicated to use, but it does require a little bit of Pythonic thinking. So histograms work, and I'll walk you through the steps of putting in a histogram, and then you can try it again on your own. Histograms, the first thing you have to do with a histogram is you have to create a plot window to put them in, just the way you do for a scientific plot. And we have in Twittit the option scientific plot histograms. And under scientific plot histograms, there is the first option, which is add histogram plot. And it's annotated, put in the start function. And the other thing that you have to remember is that if you want the handle for that plot window to be accessible easily, you need to create it in the start function in the same steppable that you're going to be using it, because otherwise it's not available across steppables. You have to global it or pass it handle. Okay. So you do add histogram plot, and it will give you something that will look almost like what we have for the scientific plots. You'll see self.plotWin. That's the handle for the plot window that you're creating, equals self.add new plot window. And in this case, that looks exactly like our old ones. But then the next lines are self.plotWin add histogram plot instead of add scientific plot. And then we give it a name and a color. And in this case, alpha is the transparency, because very often with histograms, we have two graphs that are overlaid on top of each other. And so we want there to be some transparency so we can see through them. And so really, it's not very different from what we did for scientific plots or scatter plots. The one thing we may have to remember is that the transparency is an integer between 0 and 255. So not between 0 and 100. And you can give the plot a title and label the X and Y axes. As usual, the titles have to be unique, which seems always to be a little bit odd, but the titles have to be unique. And then the plot names are the plot series. You can have as many plots in one plot window as you want. Okay. Now, histograms don't work with data point pairs. They work with lists. And so we're going to have to create a list, which will pass to the histogramming function. And so what we'll do is we will create an empty list. In this case, we're going to be histogramming the pressures in each cell, which is what we've displayed in our tracking field. And so we'll create in the step function, we'll begin by creating an empty list, pressure equals open, close square brackets to create an empty list. And then we'll loop over the cells and we'll copy into that list the value of the cell's pressure. So we now have a list which consists of all of the pressures, all of the scalar valued pressures, float valued pressures of the cells. And we'll use the append function to do that. Okay. Once we've done that, we go into the step function where we're going to put the plot up on the screen. Again, in Twittit, we have in the Twittit Python, we have under scientific plot histograms, add histogram, step function. And you need to make sure that this, in this case, is not inside a loop. You don't want the histogram plot to be inside a loop because you've already done the loop in creating the list. And there you have the plot name, which has to match what you created the data series with. The value array, which is the handle for the handle for the list, which is going to be histogrammed, and then the number of bins. Because with a histogram, you have to tell it, are you going to arrange those data into 2 bins, 4 bins, 8 bins, 10 bins, and so on. Too few bins, the histogram won't tell you very much. Too many, you're going to have either a 0 or 1 in each bin. In this case, if we're only histogramming the pressure, we'll get rid of the extra lines and just keep the first one. Okay. So do people think that they could go through and do that and add that to their code and do a histogram? So what are the steps? The steps are go to the start function, in twidit ++ pull down histogram in the Python menu and do create histogram and give it a name that's appropriate for the pressure. And then in the step function, loop through the cell. First, create an empty list, loop through the cells, and append the cell.pressure to the list. And then outside of that loop, paste in from twidit, paste in a plot histogram function. So I'll give people a chance to do that. And when you've got it and if somebody wants to show me their results, that would be great. And if people need help, please speak up.
I sent the zip.
Looking, but it's front of that. You know what I mean?
Yes.
It's always changing. Who's mad?
So if it's jumping around too much, you might update the histogram every 10 time steps instead of every time step.
Yes, the variable that tracks the flow.
Alex, is that making sense to you?
Yeah, I think I had it from before, but I'm just doing it all again to make sure I can.
I think in last week, I think Pedro gave you the code for that in the chat. This time I want to make sure people can do it on their own. If everybody could do it on your own, then we can move on. But I want to make sure everybody's comfortable creating a histogram from scratch.
Customize the appearance of the plots.
Sorry?
How do you customize the appearance of the plots?
So
Like, what are the?
one of the basic things, if you just click at it
Mhm.
and do a little drag,
Mhm.
it's free, like you freeze the X and Y rays.
But what if I want to change the colors?
So Emma asks, can you change the colors? When you create the plot series, you can declare the color. There are probably some other settings that I'm not familiar with.
Items. Where the declare function you can choose all.
Okay.
But I don't know.
There on the-
I never used it, so I don't know what it is.
It looks like it's saying the number of cells is -100. You see that?
The number of cells is negative.
The pressure, you're plotting pressure and calling it cell numbers. And the pressure,
that
yes, the
makes
pressure can be negative.
Okay.
So yes, you want to probably
Oh, yeah.
label your X and Y axes to make them sensible.
Yeah, actually, this is the code, so the default
Yeah.
is wrong.
So I just forgot to change it, and now I thought that I.
Anybody else? Jonathan, did that work for you? Alex?
Almost there.
Okay, no, I'm sorry. I don't mean to be distracting people. I just don't. Again, well, give me a thumbs up when you've got it online working. So I'll
Simulated world basically supposed to correspond to real units of pressure, or are they just these? made up of-
Simulated units of pressure.
They don't correspond to real life units. But algebraically it does. If you
So it's valid as a model, not as a.
if you give a unit of energy to energy, pressure will have units of pressure. So it's consistent. But yeah, in practice,
Yeah.
it's a-dimensional.
Right. Emma asked, how would this pressure that we're calculating here correspond to the actual pressure in the cell? And in this case, what we're doing is we're using the pressure on the cell as a surrogate for contact inhibited signaling. And so we don't actually expect that that number will correspond to a real pressure in the system. However, Pedro pointed out that if we make the energy of the volume constraint an actual energy, so if we said that the energy is so many ergs, for example, then the pressure actually would have the correct units.
I have a question. What is an erg?
So ergs are the energy unit for CGS units.
Isn't it joules?
Joules are a complicated one, but that's for MKS.
CGS.
So as usual, there are many unit systems, and they're different scale factors, just the way you can measure things in microns or in millimeters. Energy has a variety of unit names for different energy systems. Sorry, different unit systems. In general, in our world, we're not going to worry about that too much. The forces tend to be more meaningful. Forces in biological systems are normally in units of piconewtons, and if we wanted to, we could calculate what those forces are. And from the forces, you can then derive the energy scales. So I'm waiting. I'm still trying to figure out, are people okay here? Do people need more time? Santu says working on it. Okay. And you'll notice that the x-axis of your histogram will auto-scale. And it's perfectly okay for some of the pressures to be positive and some of them to be negative. That's handled correctly. Okay, I'll let people keep working on that a little bit. I want to talk a bit about using Player. Once you have a histogram, or for that matter, a scientific plot, you might want to be able to save the data that is embodied in that plot. And there are two ways that you might want to be able to save it. You might want to be able to save it as an image to use somewhere else, or you might want to be able to save it as a data set for further processing. And if you want to do that, you can do that from Player while the simulation is running or paused. And to do that, you would hover over the graph and right-click over the graph. And when you do that, you will pull up a small dialog box, and that dialog box will say View All, X-axis, Y-axis, and that'll allow you to change whether the X and Y axes are fixed range or floating range. There's also a plot option window and export. I will warn you in advance that some of the plot options can look pretty fancy and some of them will crash player. They're not all actually implemented. There's one that in principle will give you the Fourier transform of your data series, and that won't work. But the basic ones work. And so if you select export, it'll pop up a window and you can select an entire scene, and then you can export it as an image file like a PNG, TIFF, or JPEG, or you can export it as a CSV. So you could export it from there. Why don't people give it a shot and see if that works for them? Give me a thumbs up or a thumbs down if it works. We need a special icon. I guess a thumbs down would count for if it crashes player, give me a thumbs down. I do guarantee that if you try to do some of those fancy plot options, they will crash. Several of them work. For example, displaying grid lines will work. But there's some fancy signal processing options that aren't implemented. These dialogues are not from CompuCell. These are dialogues that are packaged with the package that CompuCell uses. So, was anybody able to get that export button to work, either of an image or of a data set? Emma says yes. Alex says yes. Did it crash on anybody? No. Okay, something that somehow got left out of our documentation, which I want to walk you through quickly, is that saving things from the screen is just fine when you're running it. But if you were running a batch mode or not there to watch it, you might very well want to save it from your Python. And we haven't really talked about file access from CompuCell very much. And I want to, used to be that CompuCell was really quite a bit of a problem to do file reads and writes. It's gotten a lot simpler over the years. And again, Twidit is your friend here. So these code snippets live in the Python/Python utilities submenu and also in the Python/scientific plots or menu. And so let's suppose we want to save the plot image and data set from the simulation itself. The first thing that we need to do is specify where we're going to save the simulation data. And whenever you run CompuCell, and if we were saving a lot of stuff, you might notice this because it might fill your disk up. Every time you run CompuCell, it creates an image of the simulation that you ran in the folder by default called CC3D Workspace. And so one of the reasons that it's important to have your system installed in a writable directory is that by default that CC3D Workspace gets put in the same place as the CC3D, and you don't want that to be a non-writable directory. And then the name of the folder, which will contain the copy of the simulation and its output, will be whatever you named the simulation, and then underscore, and then a version of the date. It'll have the day, month, year, and so on. And the simulation outputs by default will go into that directory along with the specification of the simulation itself. And the idea here is that way, if you needed to rerun the simulation to make sure it worked, you have exactly what you did to create the simulation in the first place. Again, if your simulations are producing very large amounts of, say, movies as output, it can be cumbersome if you're running them a lot of times. But in general, as a default, it's better to save everything and then let you delete it rather than the other way around. CompuCell provides a internal called self.output_. underscore dir, which is a pointer to that workspace / simulation name date directory. In the old days, you had to figure that out on your own. Now you have it. And so what we are going to do is create a file name, and that file name actually really will be a path name for the output plus the file name. And if we're going to be writing the file repeatedly instead of just once at the end of the simulation, then we better give, each time we write it, we'll give it a unique name. And so here I have, if Monte Carlo steps less than 50, and self.output underscore dir is not none. Why am I checking that? Well, it's possible that you clobber self.output underscore directory. And in that case, you don't want Python to crash. So you want to do a check on whether that output directory is specified or not. And then we'll define a file name. And here it's just string of path of self.output directory. So I've turned that handle for the self.output directory into a string. And then I'm calling it, in this case, the file, I'm calling it extra plots, and then appending the Monte Carlo step so I get a unique name. And then the file type here is a PNG. And then I have self.plot underscore window, my handle, save plot as PNG, and then the file name. And that file name is the full path name, has to be the full path name. The size of the image I want to specify in pixels, and that will save the image state. And that same command will work both for scientific plots and for histograms. If I wanted to save the data set as a CSV, I create a file name as well. And here, instead of save plot as PNG, I say save plot as data. And in this case, I just need the path name because I don't have to specify the size. And again, all of this is in the twidit ++ snippet. So why don't people try adding to their code after you create the histogram, the code to save either an image or the data set of the histogram, okay? And let me know if it works. In this particular case, you shouldn't have any issues with creating the files and the file access. If we were doing more elaborate file handling, I recommend, if people remember how to use try, in Python, there is a command called try that does error trapping, so that if the file open or file write fails, you can generate an error message rather than having Python crash. And so I'm not going to cover that here. You're not going to have an issue with that the way it's set up here. But in general, when you're doing file handling inside of CompuCell, I recommend using the try function to do file trapping, to do error trapping. It's pretty easy to try to read or write a directory you can't read or write to. It's pretty easy to try to create a file that already with the same name as one that already exists, especially if you're writing to a central location instead of, for example, to the working directory here. And so there are a variety of situations in which file handling can fail. And so familiarizing yourself with the try capability is a useful thing to do. There also are demos which implement all of these things. If you look in the CompuCell demos, there are demos for scientific plots that show you how to do all of this. So all of this code is in the demos that are packaged with CompuCell in the scientific plots demonstration package. So again, if people could give me a hands up or thumbs down when this worked or failed, let me know. But I know people that asked specifically to how you save plots internally from the program. Okay, Alex says it works. Now, if I want to write a general data file, things are a little bit more complicated because I'll have to do an open command to create the file. In this case, the save plot knows how to create that file as well as write to it. But in that Python utilities subdirectory, there's some file manipulation functions as well. Emma says it's okay. Does anybody need more time? People get it to work? Does anybody need help?
Actually, I'm not sure it worked.
You want to screen share it?
Let me see if I can find, I just tested it for the first time. Let me see if I can find it. This goes in the step, right?
Yeah, you put in the step function.
Let's say I want to save every 1,000 months.
So if this is syntax for saving every X Monte Carlo steps, there, yeah, it's sort of counterintuitive. It would be if not MCS percent 1000 colon or what? That's the simplest way to do something every X Monte Carlo steps. You also have the option with the steppable to call the steppable every 10 Monte Carlo steps, every 100 Monte Carlo steps, or so on. If you wanted to have a dedicated steppable to do that output. But I tend to call the steppables every Monte Carlo step and then use that mod function to the only thing you have to remember is that mod function in Python is that percent integer division. Yes, in this case, if MCS is less than 50 was just a placeholder. You could have if not MCS percent 100 and self dot output or not none.
No, actually, it worked. I just forgot that it's in that CCTV workspace directory, which, yeah.
All right. Okay. How about Alex? Did it save a picture? Did you save a picture or a data set?
Pictures. I've tried data set too.
I'm trying
Okay.
to use the tracking to see pressure, and it's telling me, okay, here are the minimum maximum pressure values. So it's getting the data, but the data aren't showing up with the scale. I don't know how to set it. It seems like it just doesn't permit negative numbers.
Okay, Emma says the tracking field is causing problems for her
I tried to add one with the pressure.
for the pressure. So sometimes, and I'm not quite sure what triggers it. Sometimes the tracking fields will fail if the values are negative.
You know what the source code is for those?
Yes, the source code. Emma asks, where is the source code?
I know the source code, all of it is on GitHub, but I don't know how to find specifically the tracking codes.
So the modules are pretty well named. So if you look for tracking field, you'd be able to find it. And if you notice a problem like that and you can document it, please do report it. We can open a ticket for it.
Scalable variable and then like adjust that.
Anybody else? Has everybody got that basic save work to work?
Just in the configuration for
Okay. Okay. The next thing that I wanted to do here was come up with something that is one of the few things that's not in Twit. Probably should be. We need to add it to Twit, which is text boxes. So this is not something that you necessarily will wind up using a lot for yourself. But if you're going to be writing an application that's going to be used by other people, the fact that the text that you display is output not to player but to the console can be an issue. So you might want to be able to, for example, if you're going to make an educational application or something that users are going to be interacting with, we're going to be developing sliders where we can change the values of parameters while the simulation is running. And that will pop up as a box inside of player. We'd also like to be able to display things on the screen inside of player. And player actually does have an option. At the bottom of player, there is the text box where error messages pop up. And in the settings for player, there actually is a button that says direct output to that box. And it doesn't work. And so instead of doing that, what we have is an option to pop up a message or dialog, a message box inside of player, just the way we can pop up a plot window. And it's called add new message window. And so in the start function, we would type self.message window. That's just a handle, so you can give that any name you want. Equals self.add underscore new underscore message underscore window, parenthesis title, and then the title of the window. And then to print into the window, you do self.message underscore win, the handle, dot print, just like the normal print function, and then in quotation marks, a string that you're going to print, and then you have a number of options. You have a color, and you can have a style, bold, italic, or underline. And you may also want to erase the contents of the window, self.message underscore window dot clear. So why don't you, everybody got that? Why don't people try that? Just not very exciting, but just add in your step function something that's going to display the step number in MCS, add the number of cells that are present in the simulation every 10 Monte Carlo steps. Okay? And remember that in the start function, you have to create the message window. And in the step function, then you have to create a line that does a print to that same message window. The thing you want to print with a style and a color. And in this case, I'm only printing MCS, but I'd like you to print the number of cells too to remember how to do that. So that's a little mini exercise. So see if that works for people. Give people a few minutes to do that. Again, a thumbs up when you've got it working would be great. And if you have a problem with it, let me know. Again, this could be used in a variety of ways. For example, if you have a simulation that's going to be an educational simulation, you might want the simulation to explain pop-up text that says what's going on in the simulation at different times, or it might pop up instructions to the user about how to change parameter values. Or it might pop up conditions or problems that happen during the simulation. It's not a very sophisticated text interface, but it allows you to communicate with the user while you're running the code. If anybody has it working or not working, let me know. Or if you need help with that, let me know. The only thing fundamentally that may not be obvious, Alex, it worked for you? Good.
Yes.
William, what about you? When I'm developing sort of demos or educational apps, I use this feature a lot. Again, it's not very elegant. It's a very basic dialog box, but it does give you the ability to display things that the user will notice while you're running. One thing that you may find if you print a lot, and that's why I gave you that mentioned that erase function, is that the content of that window is buffered. And so if you put too much information into that window, you can actually crash player. It can overflow the buffer. So erasing it once in a while isn't a bad idea. Did anybody else get it to work? Pedro? No. Pedro's not interested in trying that one out.
I'm getting a MCS is not defined. I'm assuming I'm not attributing something where I should be.
Well, let's take a look. Do you want to screen share and let's
Yeah.
see what we got? Sorry, let me stop the screen share.
Either he doesn't have a variable MCS on the declaration of the function being passed, or he's using uppercase, maybe MCS applicated.
Okay, let's see here. Have to relay out my screen. For the people who are in the room, they're not going to be able to see your. Let's see. Set this up.
I'm sorry. I'm gonna do.
What is the line? 38?
Here,
Oh,
zooming in.
here. The answer. Sorry.
I'm sure it's very simple.
Sorry, Kyle. Sorry, Kyle. sorry, so the issue here, this is an example where the pasted text has to be edited. Your style has to select which style you're using.
Okay.
You can't have it be bold and italic or underline. You have to pick one of them.
So
Or
if
you
I
can
just
get rid of the
pick
style
bold, for
entirely.
instance.
Yeah, that should be that. Try that.
It's not gonna work. Because there's no MCS and stuff.
Oh, you're in the start function.
Yeah.
Okay, so sorry. The other thing is
Oh wait, I'm in start. I should be in steppable, correct?
step. Yeah, you have to add the display to the step. So add new message. Add new message window goes in the start function.
So
This is good. So put but the line 38 where you do the print, that has to be in the step function. There you go. Try that. Oh, still crashed on you? MCS is not defined. Let's see what line it says it's happening in. Scroll down. Line 38. No, it
38.
didn't save. You
Yeah.
have to, you didn't do this. It didn't replace. You have to save.
Yeah.
Always hit save all okay, now try again.
Oh, it's indentation error.
That's a, that one,
That was just nested. There we go. And then a lot of
Okay.
steps very
Right.
quickly.
Yeah. Okay.
Thank you.
Okay. Thank you. Thank you for being willing to go over that with us. Anybody else have an issue that they want to go over with that? As I say, most of the time we have the Twit code snippets. That one didn't make it into the code snippets, so we have to that one we have to do on. Okay. There is a demo for it though, so if you get confused, there is in the demos. There is a message window demo. Okay, so I'd like to come back to concentration fields of diffusion now. Remind you that chemical fields and concentrations are an approximation to a reality where we have particles, which are atoms or molecules, and that concept of concentration depends on what we call a mesoscopic scale. That is, there's a volume in space that's small compared to the volume we care about, and large compared to the typical separation between your particles so that you can write a density. And as Pedro showed last week, you have many different kinds of concentration fields that are of significance in biology. Critical ones that we're going to run into all the time would be oxygen, glucose, growth factors like vascular endothelial growth factor A, VEGF-A, if we're talking about the immune system, cytokines, and so on. And in the modern world, it's a little harder to visualize chemical fields than it is to visualize genes because the molecules are small, typically. But there are increasingly good ways of visualizing chemical concentrations. Something that I want to just allude to very briefly is that in reality, there are multiple ways that chemicals move around in physical and living systems. There are the normal ones that happen in physics, and then biology has some special kinds of transport that don't exist in non-living systems. And Compusol is not going to support a lot of these, but it's important to at least be aware that they exist and be aware that we're not including them and think about what the meaning of not including them is. In particular, there are three main kinds of transport in biology. You have advection, active transport, and diffusion, and Compusol supports diffusion. And to a very limited extent, you can do advection or active transport, but it requires more work than we're going to do right at the moment. One of the things that we're going to run into, in particular with advection, is that in CompuCell, the chemical fields are carried in lattices, in arrays, and those arrays are separate from the cells, and therefore when cells move, they don't move the chemicals in the fields. And that means that by default, we don't have advection. Whereas in the real world, you automatically get advection. So for example, suppose that I have a fluid flowing in a pipe and I put a drop of ink in that fluid. When I have pipe flow, the flow in the middle of the pipe is faster than the flow at the boundaries of the pipe. And so that ink drop gets carried by the flow, and it also gets stretched because the flow in the middle is faster than the flow at the edge. And so you get something that looks like what I show here on the right. So chemicals that are dissolved in a fluid that's moving are carried by the movement of the fluid. If I have a chemical attached to a solid and the solid moves, it's also carried by the movement of the solid. I don't get typically that advective movement for free in CompuCell, and that kind of movement can lead to very complex reorganization of chemical concentration. And so leaving it out, which most computational models do most of the time, unless you're modeling vascular transport, actually can have relatively major consequences on your simulations. And so this is something that one has to do quite a bit of work around. One of the reasons that we don't implement advection is that actually doing fluid flow is quite expensive. Solving the fluid dynamics equations is hard. And so if you really want to solve fluid flow, you have a very heavy computational burden. There are software packages which couple fluid flow to multicellular models, but that tends to be pretty specialized because the way you do that coupling is pretty dependent on the problem you're solving. The other thing that you have, which is very biological, which is specific, is active transport. And there are multiple kinds of active transport. At membranes, you have ion channels that cross the membrane. And some of those ion channels are passive. They basically act as just holes. But a lot of those ion channels act as pumps. And you have things like symport pumps, where two molecular species move together through an ion channel, or one of them actually acts essentially as fuel to move the other one across. Antiport pumps, where you move actively in counter direction to each other. You have ion pumps where ATP is used to move a chemical. And in some cases, those pumps are pretty elaborate. Cells continually have to pump water out of the cell because of osmosis. Water is continually being moved from the extracellular environment into cells. If cells run out of energy, they have to bail, essentially, like a leaky boat. If the cell runs out of energy, runs out of ATP, it'll swell up and eventually burst. And so when cells have ATP depletion, there's what is called hydropic swelling. You'll see the cell will expand like a balloon and burst necrotically if it really runs out of ATP. What's interesting is that the same pump that does the water pumping there is also used in reverse to synthesize ATP, which always seemed to be a bit of magic. I've never quite understood how that works chemically. It doesn't seem to me that it should work. But we can do some simulation of active pumping. There are some packages that have more sophisticated transport relations at boundaries than CompuCell does. The finite volume solvers and that TJ's written do some of this, although we're not going to explore those so much in this course. The other kind of active transport that you get in biology, again, is something that's very specific to biology, which is vesicular transport. Cells will package particular chemical cargoes in lipid vesicles. Those vesicles will have molecules on their surface that bind them to particular molecular motors, things like kinesin or dynein, and then those vesicles will be actively transported along the microtubules in the cell. And so, for example, in neurons in particular, the way that the very long axon and dendritic fibers are maintained depends almost entirely on the presence of molecular motors that move the molecular species from the soma of the cell where they're synthesized all the way down those very long fibers to the ends of the fiber. If you depended on diffusion to do that, you couldn't have neurons. Wouldn't work. There's also endocytosis and exocytosis, which are involved in both normal function and in disease. In particular, endocytosis and exocytosis are very important for infection. One of the ways that viruses get into cells, not the only way, but a common way, is for the virus to mimic something that the cell normally internalizes. It binds to a receptor that causes the cell to take the virus inside. So a mechanism that the cell normally would use for nutrition, for example, is hijacked by the virus.
Can you hijack it for good purposes?
Kyle asked, can you hijack endocytosis and exocytosis for good purposes? And the answer is absolutely yes. In synthetic biology, very often you will build systems which either synthesize or absorb chemicals in an engineered way. We're going to be focusing on diffusion. Diffusion is the passive one of all of these things. Doesn't involve active transport. Diffusion is simply the movement of chemical from regions of high concentrations to regions of low concentrations. It's fundamentally, it doesn't require the addition of energy. It happens automatically due to the second law of thermodynamics and the increase of entropy. Fundamentally, if I have a high concentration of chemical on the left and a low concentration on the right, and each particle has an equal probability of moving to the left and to the right, the probability, if I look on the right, where there are no particles, the probability of a particle on the right moving to the left is 0, and therefore the flux from the left to the right, from the right to the, I'm one of these, I'm a person who cannot tell right from left, so I have to look at my hands to be able to do that, so I should be a little careful before I say something. So on the right, there are no particles. On the left, there are many particles. If the probability of each particle moving to the right or to the left is equal, and I ask what is the net flux, if I look on the right-hand side, the fraction of particles moving to the left is 0 because there are no particles. If I look on the right, that's what happens. If I look on the left, in towards the left middle, there are particles that are moving from the left to the right, but there are no particles moving back because there are no particles to move. And so there's a net flux from the left to the right. And that's how we get equilibration of gradients, the dissipation of gradients. Diffusion primarily is thought of as something that happens at the molecular scale, but it also applies at larger scales as well. Anything that moves randomly will diffuse. If I look at bacteria or plankton in a fluid, or even animals in an environment, if I start out with a large concentration of zebra in one spot, they'll gradually spread out Of course, they also will actively move and maybe cluster again under particular circumstances. And it doesn't have to be molecules. Heat also diffuses. Thermal activity also diffuses. Something that's going to be important is that diffusion is a short-range process, and we'll come back to what that means in a few minutes. The basic physics of diffusion is pretty universal, but the rate of diffusion of the same chemical species will depend on the environment we find itself in. It'll depend on what other molecules are present, whether there are membranes in the way, whether there's crowding, what the viscosity of the fluid that they're operating in is, and so on. I'm not going to go through the mathematics in detail, but we can come back to that if we like. We'll define the concentration as the number of particles in a volume here. So the number of particles in a volume XYZ. These images are taken from this wonderful book, The Physical Biology of the Cell, that I've recommended before. If you want to dig into the mathematics of diffusion, I strongly recommend that textbook. It's a great place to learn these things. If I look at the boundary of my cube, and I want to ask what is the flux across one of those boundaries of the cube, the flow rate, given the argument that I just made about jumping to the left and to the right, is proportional to the concentration difference at any point. So the net flux at any given point in my volume is a diffusion constant. Where the diffusion constant comes from is effectively the rate of jumping of the particles times the gradient of the concentration. So if my concentration were uniform, the gradient would be 0. There would be no net flux. The steeper the gradient, the faster the flux. It's important to notice that diffusion has units of distance squared per time, and that's going to be why diffusion only works over short length scales. If I think about my box and ask how much chemical do I have inside of my box, then I can use a simple vector calculus relationships that will tell me that the net change in concentration of my box, inside my box, is the divergence of the flux. This is a Gaussian integral over the surface of the box. And so I then can derive the diffusion equation. If the diffusion constant is not constant in time, then I'm going to have the divergence of the diffusion constant show up, and I'll get two terms in my diffusion equation. Most of the time, people assume that the diffusion constant is the same everywhere, or at least changes very slowly, so you can neglect the divergence of the diffusion constant. And if you assume the diffusion constant is constant everywhere, you get that the change in concentration is equal to the diffusion constant times the Laplacian of the concentration. Now, CompuCell, as we'll find out a little bit later, allows you to have non-uniform diffusion constants. You can have the diffusion constant d be different in different places, but it will still solve the diffusion equation as if the correct diffusion equation were d del squared c. It won't do the second term. It'll assume that the change of concentration is slow compared to everything else. And so you should be aware that that solution is only approximate. Okay. Now, again, it's important to remember that in biology, unlike, say, diffusion in water, where water has basically the same diffusion constant everywhere, the diffusion constant is not the same everywhere, and it may not even be a scalar. It may actually be a vector or a tensor. So, for example, diffusion in water is much faster than diffusion in cytoplasm, which is pretty viscous. Diffusion across membranes can be very slow, and it can depend on the ionic, both the size of the particle diffusing across the membrane and the ionic properties and polarization of the particle. Some molecular species stick to chemicals in the extracellular matrix, and so they have very, very slow diffusion constants, effective diffusion constants in the presence of extracellular matrix. There are molecular species that are, many of them, that are what are called membrane bound. They have tails that stick to the membrane of the cell, but still they can move freely within the membrane. And so then you'll get two-dimensional diffusion within the membrane, even though you won't get much diffusion of the chemical across the membrane. That's something that CompuCell doesn't implement. Morpheus implements that, for example. When you have fiber architectures and the fibers are anisotropic, so that the fibers are aligned with each other, as for example happens in scar tissue or in muscle, then you very often will have situations in which the chemical will diffuse faster along the direction of the fibers than perpendicular to it. On the other hand, if you think about cells as active agents, and you have cells moving randomly along an organized fiber structure, you'll find that it goes the other way. Actually, I should say that it's going to be the same also. If you have fibers that are aligned with each other, you'll find that cells move preferentially along the direction of fibers and not perpendicular to them. And so in those cases of anisotropic diffusion, life becomes more complicated. We're not going to implement those here, but it's important to be aware that that's possible. And so when you build models, you need to be aware that you may be making simplifications about that. Again, the chemical fields in CompuCell are simple arrays of floats. They could be positive or negative values. Nothing very complicated there. CompuCell also has a vector field object where you can have triples of floats if you want to represent orientations. The diffusion solvers don't work with that. There are many ways you can actually numerically solve the diffusion equation. There are very fancy ones that are very computationally efficient. The problem with those is they tend to be restricted to particular geometries in particular situations, like the diffusion constant has to be the same everywhere in the simulation, or the shape of the simulation has to be a cube. The simplest way to do diffusion is to, at each point in space, average the values over the neighborhood. That's called the Gaussian kernel integration. And that's the one that CompuCell uses for most purposes. It is not the fastest way of doing things, but it has the advantage that it allows you to have non-uniform diffusion constants. Again, there's a whole universe of fancy diffusion solvers if people are interested in that. There are quite a few diffusion solvers packaged with CompuCell. Well, quite a few may be an exaggeration. There are several. There's a steady state diffusion solver which tries to calculate what would happen if you run the diffusion for a very long time. That is useful for chemicals that diffuse very rapidly. In practice, in biology, that means oxygen and maybe ATP and glucose. It's also quite expensive computationally. And so you typically want to avoid using it unless you absolutely have to. Diffusion solver FE, which is the one that we're going to use most of the time, it used to have a GPU version. People can see if that GPU version works with their computer or not. I'm not confident that it's going to work anymore. When GPUs evolve, the GPU solvers have a tendency to break. And then one that we probably won't use so much here, but is a lot of fun, is the reaction diffusion solver, which allows you to have multiple chemical species interact with each other. And there's a whole zoology of patterning of reaction diffusion equations that you can implement in CompuCell. That's actually a good class project if you're interested. And there's the new one that TJ has written as well. If we're going to go into Wizard and create a chemical field, I believe you did this last week, you can use the Wizard dialog box. We typically have been skipping the chemical field object dialog, but in the Wizard, you would go to the chemical fields, and this time you would create a field. Here you could say type oxygen. You select the solver you want to use. The default is diffusion solver FE. You hit add. And you can add as many chemical fields as you need to your problem. That's it. If we look inside of the XML, we're going to see that we have a steppable implemented in the XML, which will tell us what the diffusion solver type is. Here, diffusion solver FE. Then the name of the chemical, here, oxygen. And then you'll see diffusion data, and then it'll see field name oxygen. And I'm actually not sure why we have to have the name oxygen specified twice. And I haven't played around with what happens if you have the names be inconsistent with each other or you leave one of them out. The next thing you'll see is a global diffusion constant, which is the default diffusion constant in the system, which corresponds to that D in the diffusion equation. A global decay constant, which we haven't played with yet, which we'll come to in a few minutes. And then you have some additional options. One of them is to specify a simple arithmetic form for the initial concentration that you're going to use in the simulation. You're allowed to read the concentration from a file, which could be useful if you want a complex initial condition. And that's it. You can also, of course, specify all of these things in Python. People said that they had found the idea of boundary conditions a little bit challenging. And so I wanted to come back to boundary conditions, give people an option of asking some questions about it, maybe doing one or two simple exercises about it. In reality, our simulations are always going to represent some finite volume of space. Now, in certain kinds of experiments, for example, if I'm in a test tube, there really is going to be, or a petri dish, excuse me, a petri dish or test tube, there really is a wall. There's a glass wall or a plastic wall at the edge of my test tube where my living system stops. There's a layer at the bottom where I might have a substrate, and a layer at the top where, for example, my fluid ends and I have air. But in most of the simulations of living tissues that we're going to be representing, the simulation represents a window onto a tissue that continues outside of the field of view. And this represents a problem for simulations because in reality, the tissue doesn't stop. The tissue continues and there are more cells outside of the field of view. The problem is that I don't know what those cells are doing, and so I don't know how to simulate them. And so if there's a hard wall that's at the edge of my universe that I'm simulating, things are easy. But if I'm actually trying to simulate a patch of tissue, I have a problem. I don't want to create discontinuities at the edge of that patch. And in that case, we typically use periodic boundary conditions. Why do I use periodic boundary conditions? Periodic boundary conditions essentially make my tissue infinite by rolling it into a donut. And so getting rid of the edges. Now, periodic boundary conditions have some problems. If I have a gradient from one place to another, then I'll get a discontinuity of my concentration when I jump across that periodic boundary condition. They also mean that calculating things like distances across the edges is complicated. But they're also probably the most common boundary condition we'll use for many purposes. Constant flux boundary conditions mean that at the edge, there's a flow of chemical in or out at a constant rate. That can have some perverse consequences. If I have a constant flow inward, I basically have a hose that I'm pouring chemical in. That means the concentration can go to infinity. If I'm pulling chemical out at a constant rate, then the concentration could go negative, which doesn't make sense either for chemical concentration. Constant value boundary conditions say that I specify that the concentration at the boundaries is a fixed amount. Those tend to be better behaved. They won't give me negative concentrations or zeros or infinite concentrations. And they would correspond, for example, to what happens if I have a blood vessel. The blood in the blood vessel typically will have an oxygen level that's pretty constant. And so to assume that the concentration is clamped at a particular oxygen partial pressure is not unreasonable. Not perfect, but not unreasonable. But depending on what kinds of biological phenomena I'm trying to model, I might have to use different combinations of these. For example, endocytosis and exocytosis. Exocytosis in particular, the cell is dumping chemical into the environment. The rate at which that chemical is dumped depends on internal processes in the cell, not on the concentration outside the cell. And so in that case, constant flux would make sense as a boundary condition. So this is a nice slide that I believe Pedro did originally, where we start out with the chemical source here. And if we have periodic boundary conditions, we see the chemical goes to the edge and spreads around the edge. Again, think of the field as being wrapped both around the x direction and the y direction like a donut. And so there is no edge. There is no edge effect in this case. If I say that there's a constant derivative and the derivative is 0, that's equivalent to what are called no flux boundary conditions. That effectively is equivalent to having a rigid wall that either absorbs, that doesn't absorb the chemical and doesn't secrete it, also known as reflecting boundary conditions. And in that case, the derivative is 0. That means the concentration can't change at the boundary. And I'll see that in this case, the concentrations get to be pretty high at the boundary. If I say that there's a constant value of 0, now I have perfectly absorbing boundary conditions, and we'll see that the value is 0 at the boundary. You'll notice that the concentration is not only changed right near the boundary. The concentration is different over a large range of the simulation. If I go to the upper right-hand corner, and I have periodic boundary conditions, I have chemical. If I go up to the upper right-hand corner where I have constant derivative, I don't have any chemical in that corner. If I go to the upper right-hand corner of constant value, I have no chemical. And so what happens even quite far from my source and quite far from the boundary can be different depending on the boundary conditions I picked. So it's quite important to think carefully about what is appropriate in my model. And we are often stuck because the actual boundary conditions that are present in the system, we don't know when we have a field of view problem. And so we have to be aware of that. These are all going to be approximations, and the quality of those approximations will be something that we have to evaluate.
You have constant value that's negative, or does it not make sense?
So Emma asked, could we have constant value at the boundary and set the constant value to be negative? Well, it depends what we think that the chemical field, well, I say chemical field, what we think the field represents. if the field represents chemical concentration, it doesn't make sense to have a negative concentration. On the other hand, the field could represent the electrical potential, and the electrical potential could be positive or negative. So whether fields have to have positive values or not really is a question of how we interpret the field. Numerically, there is no reason we can't have negative and positive values. The diffusion equation works for negative values as well as positive values. The one thing that you can't have is a negative diffusion constant. If you make the diffusion constant negative, you've turned your diffusion equation into a wave equation, and you get oscillations.
Don't you just get like concentration of stuff like concentrates?
So no, if you run the diffusion equation with negative diffusion constant, you get a wave equation. You get if you think about it in one dimension, d squared x by dt equals, dx by dt equals second derivative of x with respect to position. If you have a positive d, then you get an exponential, a decaying exponential solution. If you have a negative d, you get e to the minus I omega times t, because the number in the solution is the square root of the diffusion constant. So if the rate of decay is imaginary, then that's an oscillator. You get sine waves. That's a very physicsy thing to talk about. In physics, typically you go the other way. You learn the wave equation first, and then you learn the diffusion equation. That happens. But I think really the best way to understand the boundary conditions is to play with them. To specify boundary conditions in CC3D, you do that in the markup language. You have a variety of options in the diffusion solver. For boundary conditions, you could control them for x, y, and z separately, and for the left side, the min side, and the max side separately. You have a choice. You could make the boundaries periodic in x, periodic in y, periodic in z, with a periodic tag. If you say periodic, then by definition, that applies to both the left and the right-hand side. You can't have it periodic on the left and not on the right. So that one, you only have one option. But for constant derivative or constant value, you have the option of being independently specified on the left and the right. So min specifies left, right specifies, max specifies right. Constant derivative, typically we'll use 0, which is no flux. But you could, if you have a source that's being pouring in at a constant rate, you could have a source. Making a negative value is a little bit strange. And you have to think about what direction the flow is. So sometimes you'll see that the value has to be the opposite sign of the one you expect. Constant value is a simpler concept. Often it will be 0. That will be absorbing boundary conditions. Sometimes it will be a fixed value when you have a source like contact with the atmosphere for oxygen or contact with a blood vessel for glucose. The default boundary conditions that Wizard gives you when you create chemical fields are not the ones you want. And so you're always going to have to edit those. So why don't people create a simulation with one chemical field and set the global diffusion constant to 1, the global decay constant to 1. We'll use the initial concentration expression option, and this will just give us some concentration. And we're going to make it just equal to 1. So the default is x times y, just to tell you that you can have spatially varying concentrations. But you're going to make the value 1. And then to begin with, make your axes periodic in X and Y. And run that simulation and tell me what you get. And make sure that the global decay constant is set to 0.0. Right. You didn't do this exercise, Pedro, did you last week?
This specific one,
Okay.
we did the linear gradient.
Okay, so if you did linear gradient, you jumped ahead a little bit, but if people found the diffusion, the boundary conditions confusing, I think it's worth going back and just playing with these for 5 or 10 minutes to get some familiarity with that. So is it clear what you need to do? So go into Wizard. You don't need to create any cells at all. Create one chemical field. And then go into Twit, go into the XML, edit the chemical field. So the global diffusion constant is 1, global decay constant is 0. Uncomment initial concentration expression and make it 1 or 1.0. And then set periodic boundary conditions and run it. And see what the chemical field does. And I'd like somebody to screen share when they've got that working. What did people get? Emma says she got a boring field with a concentration of 1 everywhere. That's right. How about other people? Jonathan, what happened to you?
I am still working on it at the moment.
Okay.
I'm getting a gradient, so I need a gradient of 0 essentially, so I just need to change it to be 1.
So if you're let me maybe then it's worth screen sharing what the code should look like. Well, possible code.
Okay, here, let me get my screen up then.
You want to screen share it? Is that no?
I could if you'd like me to.
Sure, that would be fine. Let me find my. Okay, go ahead.
Sure.
Okay, so there are a couple of things here you might want to do. First, you might want to get rid of, in your XML, you might want to get rid of the cells. So don't wait at the bottom of your simulation
These.
where you have uniform initializer, you might want to get rid of that. Because you don't actually need any cells at all in the simulation. So you could just get rid of that whole steppable there.
Just this entire thing?
Yep.
Just delete it out.
Delete that. Good. And now let's look at the boundary conditions. You have a couple of things. First here, initial condition in line 45, your initial condition is X times Y, and you want that just to be the number 1. 1.0 probably. Better.
Oh, yeah. Maintain it.
Okay, try saving that and run that and see what happens. Okay. And now now you have, let's look at what your boundary conditions are. Why don't you read off in lines 56 through 50 through 60, whatever. What are the boundary conditions you've set? Tell me what they say.
Well, this needs to be 0 and 0, I'm assuming. But so
well, try that one. Let's see what you get with that. Why don't you actually read off those lines? Tell me what they say.
So plane axis equals X, constant derivative plane position minimum, value of 0, constant derivative plane position max, value of 0. Whereas Y, so essentially this needs to be 0, 0, and 1, 1.
Well, okay, what will that do? What does constant derivative do if the value is 1?
For which one.
Any of them.
It sets it at that specific integer.
So why don't you look at the picture that you've got? Is there chemical coming in on the left or the bottom?
It's coming in at the very, very top.
It's coming in at the left and
Top right corner.
left. It's coming in on the right-hand margin and the top margin.
Mm-hmm.
So what are the margins? It's max, and you set value equal to 1. A constant derivative of 1 means that you have a constant flow inward of chemical.
Okay.
So setting the value max, constant plane position max means the right-hand side and the top. Plane position min means the left-hand side and the bottom. So why don't we see which is which? Why don't you make the first one in the X, it's hard to remember which way is X and which is Y. So why don't you make the X1 0, 0? So we can identify which is which. Now run it.
All right.
Okay, so the top is Y max is the top boundary. X max is the right boundary. X min is the left boundary. Y min is the bottom boundary.
Yeah.
Okay. And what you've done here, if you look at line 66, is it? You say max value equals 1. You've got a source of chemical at the top. That's what constant derivative of 1 means, that you're pouring chemical in at a rate of 1 unit per Monte Carlo step.
Okay.
Okay. So now if you and notice the maximum values in the color bars, the maximum value here is what? I can't see it the way it's displayed.
Oh.
But you can see there the maximum value is, should say at the bottom it'll also say maximum
14.8.
14. So it's 14. Because you're continually pouring chemical in at the top.
Yeah.
Okay. So what if you made it 0? What if you made all 4 of the boundaries 0? You're doing the whole exercise. What I'd asked you to do was set the boundaries to be periodic, and you've jumped ahead a little bit. Let's try that. Let's try running that with all the boundaries. Okay, so now the values are 0 everywhere. 0 boundary condition, 0 derivative boundary condition means that there's no flux. Chemical doesn't enter and doesn't leave. It's like a glass wall that keeps the chemical inside, but doesn't change the value. Now try making the boundaries periodic. And the best way to do that is to block comment lines 57 and 58. So select those and then use edit and block comment. And then block uncomment the periodic boundary condition. Just periodic. You don't want to do the one with, okay.
Just periodic. Oh.
Okay.
Oh,
And in fact, you'll find, now run it and see if it looks different. The answer is going to be,
hopefully.
do you think that will be different or not?
I would assume not, since we're not actually changing anything at the moment.
Because the concentration is constant, the periodic or no flux doesn't matter. If the values were different, then periodic or no flux would matter. So now you could try making that second one. Now you could put in a source. For example, on one of those, make it 1 again for the plane position.
So if I did minimum of 1, that would pump everything in from the base, correct?
There you go. Yeah. Okay. So
I guess.
Go ahead.
Or is it actually? It's not pumping in because we're still having constant.
Yes. So this
up here, so it's diffusing from the bottom.
Let's see what happened here. Max min is okay. Actually, is this still color? Okay, so one thing that's happened here is that we've got fixed color bars. So one thing you might want to do is go to the options in your player.
Mm.
Configuration. There you go. And where it says field, a chemical field, you've got fixed values, so uncheck the fixed. You see where it says
Which
min
one?
and max,
Oh,
uncheck fixed.
it does not like me. Everything's grayed out.
That's interesting. Why is it grayed out? Let's see. That's different.
Not actively running, is it? No.
Did the simulation end?
Yes, it should have.
No, it should be running. Hit run. Leave it running and go back to that option.
Okay.
Undo it. Now it's okay. There you go.
Oh, and then now we have it
Let's see.
updated.
Now it should auto scale. This is what I was saying about the direction of flow.
Hmm.
A
So
gradient
now we're
of
getting a
1 on the
minimum of
bottom
negative.
is pulling chemical out. So
How
the,
would we
the fixed,
have a negative?
because
Is it a
the fixed derivative is relative is specified relative to the coordinate system. So
Okay.
a one is pushing chemical up, a one is pushing chemical down.
So it's the actual difference that's being
Yeah.
presented in the scale on this left. Okay.
So a one pushes down and to the left. So if you make it -1, then it'll be a source.
Okay.
So it's that one is a little bit complicated.
So this is changed to -1.
Right. So -1 on
The opposite.
on the bottom behaves like 1 on the top. Now make it 1 on the top. So where you go max, now make it 1.
And then this one back to 0.
Well, why don't you leave it -1 there and 1 there so you can see that they're the same. Good. Okay, try that. So now stop that. Now you'll see, because the flow is in opposite directions, right? The flow from the top is downward, the flow from the bottom is upward. And that's why you have to have the opposite the sign has to be opposite because the flow direction is opposite. So that's a little bit of I wasn't planning on doing that one till later, but you jumped ahead and that's good. So when you're using constant flux boundary conditions, you have to be aware of that. Now, if we comment out the constant flux boundary conditions, and now uncomment the constant value boundary conditions.
Which one? Sorry.
That one there,
This
68.
one.
And you'll need to copy and duplicate that one probably, yeah.
Oh. So.
Okay.
Comment.
Good.
And then duplicate, you said?
Yeah, because you want min and max. So you'll have to copy and paste the same. Okay, and then change min to max. And now around this one, this is going to look very similar, but you'll notice the max value will not go to be large. It's going to be, the maximum value will be 1 when you run it. Let's see what happened. Why is it not? You got.
Oh, is it because periodic is still?
Let's see what happened. Why is that not working? Let's see. Plain min, plain max. Oh, you left out a close bracket.
Oh, wait,
The close angle bracket's missing,
Oh,
I think. I can't. No?
right.
It'd be right. Try that again. Run it.
Save. Oh,
Huh. Very interesting. Pedro, do you see the problem? It's a little hard for me to see the text because it's pretty small.
Can you control plus?
Oh, you're, yes, no,
Yeah.
okay, there.
What is going on here?
What is in what's, get rid of that the backslash in line 70.
Oh yeah, there's a backslash instead of a equal sign, I think. No, I think it's
The closed brackets missed, no? It looks okay.
oh yeah, right, yeah.
That looks all right to me. Let's save it and try it again.
Oh, it is right. But he put so see it's from 1 to 1 here.
Oh, because your values are all one.
Yeah.
So it's correct.
So it's working. Just
So make the initial value of 0 everywhere. Be clear. In line 45, make the initial concentration 0.
in line 45, initial 0,
Yeah.
and then
Now we'll see some.
Now we're on it. There you go.
okay.
So it looks similar to the one you had where you had constant flux, but now the maximum value will be 1.
And minimum is going towards 0.
Is 0. And eventually it'll fill
Essentially.
up and it'll all be 1. Okay, good. So let's I hope people should play with this a little bit. It does take a little bit of getting used to. There are quite a few options. And this issue of the direction of flux takes a little bit of getting used to, but I hope that those exercises give you something to play with. We've already started exploring secretion and absorption because the boundaries are secreting and absorbing in what you've done.
So what is going on? The divergence of the gradient is the Laplace.
Okay.
Are you familiar with this?
No.
Okay. So the Laplacian is the second derivative in position.
Okay.
And the second derivative in position, if you think of a parabola, which is the approximation of a concave or convex shape,
Mhm.
the second derivative is the concavity.
Oh,
Okay, so if it's upwards, cup up or cup down, right? So this is what the Laplacian is down.
okay.
If you think of this formulation, the gradient is where it's increasing.
Mhm.
So let's say you have your fluid, your concentration field here, and let's say it is increasing the center outwards. In this case, I have this shape, right? Yeah, perfect. Yeah, that's it. So you have a cup up like this, and the gradient is pointing outwards.
Mhm.
If you take the divergent of the gradient, which is pointing outwards, in this case, the divergent is positive
Okay.
because those arrows are pointing outwards, right? So it's like one step extra. So if you think of the Laplacian, which is the second derivative already, you can see the Laplacian is positive in the center.
Oh,
So may
What happens here? To look back to the equation, where is it?
okay. In the,
Yeah, if you go back to the equation.
Mhm.
So the rate, which concentration varies in time,
uh-huh,
is proportional to this shape.
Uh-huh.
If it's this shape, the field increases. If it's this shape, the field decreases.
Mhm.
That's what it's telling. Of course, so the way it's formulated conserves mass. The Laplacian conserves mass. I mean, that's how I interpret it. And if you use the Laplacian and the d outside, you are saying that the diffusion constant is the same everywhere. If you use the diffusion constant inside, now you can make it depend. Yeah. Yes, this is the version we use. This is the
and this
version
is
we
it,
use.
but it's going to vary with location.
Yeah.
Yeah.
And we can vary in location and concentration.
Yeah.
So one analogy you might want to think about is suppose that I have a pipe, or just not even a pipe, but just a surface. Suppose the surface is like this and I have water on the surface and there's gravity and the water flows down the surface. So the rate of flow is proportional to the slope, right? If it's shallower, the water will flow slower.
Wait. Maybe I just have the direction wrong. I was thinking this should be the direction of greatest increase. But if it's greatest decrease, that would make more sense.
Yeah. Chemical always flows from high concentration to low concentration.
Right. That's why I was confused here and thinking it should be negative, but
No, so because it's not negative because so it's positive like
Okay.
this, okay?
Uh-huh.
And the divergent,
Mhm.
if you take the divergent at that point, no, in this case the divergent is zero if you have a plane like this.
So suppose, let's think about it this way. Suppose that I started out with a bowl, a bowl.
Huh.
And I put water everywhere in the bowl. Under gravity. The
Mhm.
water will flow down to the bottom. Suppose I start with the bowl upside down and put water everywhere. The water will flow away. The divergence is the net flux, whether the amount of water at a given point is increasing or decreasing. And so if there's flow inward from everywhere, then I'm getting more water in the middle.
rather
If there's flow outward everywhere, then I have less water in the middle. And actually, I had originally had all the slide deck, all the slides doing all of those calculations. And I cut them to try to make life easier for people. I should have left them in probably. Easiest to do it in one dimension, probably, and then it's that don't have to worry about the vector notation.
that doesn't, I mean, as long as I can grasp the concepts, although I'll say, I think I did vector calculus, when I was college first year, so that means it's actually long time.
Yeah, well, vector.
It's not, I mean, I don't know. I should make sure I ask myself. That's-
We can think of the gradient as the force.
Right.
I guess.
Well, the gradient is essentially the flux.
So this would be the flux.
Yeah, the gradient is the flux because it's a vector.
Yeah.
And the divergence is summing the gradients up to say, is the total flow inward or outward at that point?
Yeah.
So if I have, suppose I had, if I draw a point, it's easier to do it in 2D or 1D than 3D because it just, I think, but suppose
Yeah,
I have a
doing more
point.
than
If the flow is outward everywhere, the concentration in the middle goes down.
Right, like if it were, but this makes sense to me.
No, but the gradient is always perpendicular to the surface. It's not parallel. Okay. Oh, you're thinking about the flow on, okay, my water example. Okay, that's how you're, okay. I thought you were drawing, okay, so you're doing, okay, fine, yeah. Right. So in that case, the flow is outward from the top.
Uh-huh.
It's out in every direction.
It would be.
Out.
Yeah.
Right, out.
Mhm.
And so that means the concentration is going down.
Yeah.
So-
The gradient in the top is zero.
You mean vertically.
This?
Yeah, the gradient in the top of this shape is zero.
Okay.
But at each point just near it, it's the flow is outward everywhere.
So here it's like this, but then. Oh, so it's always.
The divergence is positive.
Yeah,
The hairy surface.
if you think you are scanning the neighbors of that point, like you are in the top, and then you scan there, you see they're lower. So yes, you are like flowing outwards. But in the continuum, the gradient is zero.
At that single point.
Yeah, at that point. But when you do the second derivative, you're actually making sure the surroundings are getting lower than the point you are in average. That's why we take the second derivative. If you take on, and because of this, if you have a field like this, it doesn't change in value anywhere. If it's a plane, it doesn't matter how steep the plane is. It never changes value. You have flow, but it doesn't change value.
The flow inward and outward balance.
Yeah. Yeah. So at any point, the stuff coming out to this direction or this direction balances the stuff coming in here. And the value in the point doesn't change because it's the second derivative. The second derivative is zero. So if the second derivative is zero, this is zero, it doesn't change the time.
Well, the other way to do it would be, instead of thinking about the first derivative versus the second derivative, you drew the box. You draw a little box, and you draw a cube. You ask the question, on the left-hand side, on the right-hand side of the cube, there's a flow inward. On the left-hand side of the cube, there's a flow outward. If the flow inward and outward is the same, the concentration inside doesn't change.
It's like here.
Suppose the slope were higher on the left, on the right, and then lower on the left. If it were bent. No, well, no, yeah, okay, fine. Yeah. Now the flow inward is higher, faster than the flow outward, then the concentration will go up. Yeah, I think doing it 1D where you don't have to worry about whether it's a vector or a scalar makes it easier. Because in 1D the gradient and the gradient of the divergence are the same. And then you don't have to, then it's easier, a little bit easier to do.
Yeah, the party round in when
In 1D,
yeah.
you have your field, this is X. And then you can, for example, you have a shape like this. At this point will go down, this point will go up, this point stays the same.
Uh-huh.
That's how I visualize, and in the way I interpret the Laplacian here is the second derivative. The Laplacian here is less than zero. The Laplacian here is greater than zero.
Mhm.
So because this is greater than zero, the derivative in time is greater than zero, it's going up. And here's the opposite. That's how, then in the interpretation of the James, which is more, actually the interpretation of James is more sophisticated because it includes that formulation where diffusion constant is inside. In that sense, you are looking, oh, so from here to here, I have a decrease in concentration, so I have a flow this way. From here to here, I have also.
I think we better get going again though, because we're online people. That's the one problem with having well, not the only problem. It's a problem with having a split class.
And you can see why the boundaries play a role.
Yeah.
Okay. So one thing that I want to do here for now is another small exercise. And people could use the simulation that you just wrote, or you could use the linear gradient simulation. Either one. Which is, if you have, does everybody have the linear gradient simulation from last week? The linear gradient simulation actually, we could use the thing that we just did in about, it takes us about 1 minute to turn the thing we're using now to a linear gradient simulation. In the linear gradient simulation, make the y direction periodic and have fixed boundary conditions of 10 or 100 on the right, on the max, and fixed value of 0 on the right, and that will give you the linear gradient. Is that clear? That's what you did last week. So you don't have to reload the simulation. You can start from the one that we just did. Okay. So one thing that's very useful to be able to do is to be able to do a plot. When we look at these pictures, the colors are very appealing and the shapes are very appealing, but sometimes it's a little bit hard to understand what the actual relationship of the numerical values is. So I want to show you how to plot a value off of a chemical field. And maybe we will go through this a little bit faster, but it's quite helpful to have it. So if we're going to use a chemical field, Actually, this may be old code. Hold on a second. Yeah. So it's correct. Sorry about that. The slide is correct. I'm used to teaching this before we fixed CompuCell. So now it's easy. So it used to be an older CompuCell that when you wanted to reference a field from the Python, you had to get a field reference. Nowadays, you don't have to do that. If the diffusion field that you created is named oxygen, then self.fields.oxygen is the reference to that field. You don't have to do a reference to it anymore. It's sometimes nice to have a handle to it. So here, field equals self.fields.oxygen would be a handle, but you don't even need that. And so if we want to read the value, I just do the coordinates. So oxygen square bracket 10, 10, 0 gives me the concentration at point 10, 10. Okay. So now I'd like you to go in and create in your simulation that you have now, create a scientific plot in the start function, which is going to be the concentration of your chemical versus position. And then in the step function, I want you to step across your simulation, however big the simulation is. And I want you to plot the position and the value of the field as a function of the position. Okay. And I want you to do that for your simulation, and you should get, if you've got everything equal to 1 all the time, it's going to be boring. You'll just get a line that says 1 everywhere. If you do something like what we were doing just a minute ago with Jonathan, where you have chemical moving into the field or out of the field, then you'll see the field change. So I want people to try getting that to work. So all you're doing here is writing something that's going to walk across the image and plot the value at each point. And by now, I hope you remember how to set up a scientific plot, so I'm not walking you through the setup part of the scientific plot. Again, Twitted has it there, and the manuals have it. And one thing that you'll find is if there's a large amount of change of the value, you'll see trace. If you plot this again and again, you'll keep accumulating points. So you'll have the trace from the first Monte Carlo step, the trace from the second Monte Carlo step, the trace from the third Monte Carlo step. Each time you replot, it plots all of the previous points as well. And so if you have millions of points in your plot, it gets slow. And so in that case, it's worth remembering the self.erase all data function, which clears the plot. Or maybe you only plot every 10 Monte Carlo steps or 100 Monte Carlo steps. And that range you should make the same as the dimension of your simulation, whatever it is. And I think I have a uniform typo. It should be field, not fields, right? So this is the code that I came up with. Were people able to get that to work? If you go from the Twit menus, it'll give you the text you need. I also saved this as a demo in the demos folder. So what you should get is something that looks like this. Initially, concentration is 0, and depending on which boundary conditions you have, you'll get different time changes. But what you'll see is something like this. That if in this case I've got chemical coming in from the left, initial concentration is 0. and I'll see that the chemical will move in from the left. So why don't people try to emulate that? I'll do that in my code, and we can do that together. Here. All right, were people able to get something like that to work? So we create a plot window, call it concentration with dots. And then in the step function, we add a data point to concentration. The x coordinate is x. The y coordinate is the value of the field at position x comma 128. And we iterate for x from range 0 to 256. Did that work? Anybody need help with that?
Yeah, I need help.
Okay, Alex, you want to screen share?
Yes.
Okay, that one is, let's look at your XML. So did you create, let's see, self.plotWin equals self.addDuplot. That looks okay. Okay, what line did it throw the error on?
I think
Sorry
it's
about
33.
So 33 is self.plotWin. Oh, why is that? Let's see. Let me look at my code and see what the issue is. Self.plotWin. Self.plotWin. Can you save it and run it one more time?
Yes.
No attribute plot win. Huh. Anybody see it? Let's see what's going on. Let's go back to your Python. This has got to be something very simple. So let's see what it is. Self.plotWin add data point. Concentration, field. No, attribute.
What is the error message?
There's no attribute plotWin. Line 33. Oh, it's not creating the plot window. So it's in the start function is the problem.
But line 17 didn't throw an error.
Yeah, that's what's puzzling me. It looks like line 17 should have thrown the error. My code is on the right.
Maybe
Yeah, I
the
was
issue is
looking-
that y-axis title is concentration, which is same as the plot name.
Oh, okay. No, but the y-axis title is, yeah, try that. Try changing the, no, but I've got it the same and it's not causing a problem.
The title doesn't.
The only one that you have to worry about is the title in the self, when you create the plot window, you have to make sure the title is different.
And that's not the same name as the field.
Is the issue, what is the chemical field name?
Field one,
Oh, yeah. Field.
field one, look at line 33.
Yeah, but you have to call the field.
Oh,
I think you have to call the field.
You mean get a reference? No, not anymore.
No? Oh, interesting.
Let's see. Let's look at your XML for a second, Alex. What about other people? Did anyone else have a problem with this? Field one, that looks fine. Okay. So come back to your Python.
What's going on? Ah,
Something easy.
found it. It's very, very bad.
What is it?
It's inside the block comment.
Okay, there. Oh, yes. Okay, yes. I knew. Yep. Line 10.
it is inside of.
Those kind of things are always the hardest to find.
What? It's it's properly colored. It was inside the block comment and it was properly colored.
Okay, Alex, try that.
It's a Visual Studio Code.
There we go.
But it's, if you see, concentration here is like in gray. It's in gray. And it was inside the comment in gray. So you will never suspect.
No. Busters do it again.
Bro.
Yeah, I think that someone should change the editor so that it applies the smart color.
You can change. You can choose you can choose the deep black one, which is the best ones.
Is it in style config?
Mhm. Deep black.
Deep black. Oh my god. What about Choco? Oh no, I don't like that.
Okay, thank you, Alex. I knew those things are always the most frustrating because they're so obvious in retrospect and so hard to find. You can have the same thing happen in XML. If you paste a block into something that's already between a tag pair, it causes a mess, and it can be really hard to find. Thank you very much. Anybody else having an issue?
Should tell him about the color settings that can make sure you see the block.
Yeah. Yeah, you can show them, James, how to change the color, the syntax color.
All right, let's see. I'm not actually sure. Let's see where.
Configuration.
Config. Okay, let me make sure I'm screen sharing this properly.
Sorry. That's not helpful. Oh my god.
Okay.
Change it to
Yes,
Comic
Comic Sans, yes.
Okay. So the comment was that the problem here was that the block comments, can people online see the editor? Okay.
Yes.
The block comments are not shown in a separate color. And so it wasn't obvious that Alex had pasted his commands inside of the comment. So go to configuration,
Configure. I think.
configure, style,
Editor theme,
editor theme.
anything.
All right. Boy, Hello Kitty sounds dangerous, huh? But it's still, but the block comments are still not.
Just the deep black one is the nerd default style. Deep black. Still not. I mean, but
But the block comments are still not.
I have to click OK.
Let's say.
But at least, and I can show you that.
I personally use Obsidian, and that one does have a change.
All right.
Oh, good. Yeah. looks good.
Yeah, there you go. That's what you want. Yeah,
Good.
good
Yeah.
choice.
It's still there, but they're a little more gray.
Yeah, but at least it's grayed out, so you know that it's not the same as regular text.
You know, I can do it.
All right, well, thank you, guys. That's I learned something there. Thank you. All right. Okay, so one of the things that you'll see is that initially the concentration moves across from left to right quite quickly. And then as time goes on, it slows down. And if I ask the question, how long does it take for this bar to get to a given position? After about 120 Monte Carlo steps, that green has gotten about 11 voxels to the right. At 2,500 Monte Carlo steps, it's gotten to 50. And so this is telling us something fundamental about, hold on a second. Let me say that again. Our screen share. So people, can you see the screen now? Because the screen, I'm sorry about the screen share being messed up.
Yes.
Okay, so what you should see is the chemicals coming in from the left to the right. If I look at the time it takes for that green bar to get to a given position left to right, what I find out is that about 100 Monte Carlo steps, that bar is about 10 voxels wide. If I go to 2,500 Monte Carlo steps, that bar is about 50 voxels wide. So the width of the bar goes like the square root of the time, not the square of the time, and not the time linearly. And if I want the chemical to get all the way across 256, I have to wait for 256 squared Monte Carlo steps, which is quite a long time. In this case, in the simulation I've showed on the screen, that's 100 by 100 instead of 256 by 256, so it's a little faster. And so one of the things that we will find out, maybe not today because we're going to be may run out of time, is that the diffusion time goes like the square of the distance. The time that a diffusion takes to go a given distance goes as the square of the distance, not linear. So diffusion slows down rapidly as things get large. That's why diffusion is an efficient process over short length scales, like the size of a cell, but not over long length scales like the size of your body. And that's why your body has vasculature in it. Diffusion can take care of chemical transport over distances of tens or hundreds of microns to a millimeter maybe, but it can't take care of transport over longer distances. You need active transport for that. Before we get to that, I just want to mention a couple of other things. There are a couple of other useful field manipulation methods that you will want to know about. Those are also in the chemical field manipulation menu in Twit. It's very useful to know what the maximum concentration of a field is, field.max, the minimum value, field.min, and there's also one that'll tell you what the total amount in the field is as well. When you want to scale things or know what's happening in your simulation, being able to pull those averages is quite helpful. So coming back to this concept of diffusion time, the time it takes a molecule to diffuse a distance L is approximately L squared over D. Now, depending on the dimension, whether you're in 1D, 2D, or 3D, there's a prefactor that's different. So there may be a factor of 2 or 2/3 going on. But diffusion slows down very rapidly over longer distances. Now, if I have potassium in water, the diffusion constant is about 2,000 microns squared per second. That's about as fast as things get. Oxygen is comparable to that. GFP, green fluorescent protein, which we use to label things in living tissues, the diffusion constant is about 7 microns squared per second, and that's typical for proteins, small molecules and proteins. The diffusion constant of on the order of 7 microns, 10 microns squared per second is typical. DNA is a big molecule, and the bigger the molecule is, the slower it moves. It has a diffusion constant of about 5 × 10⁻⁴ microns squared per second. If I ask how long does it take for oxygen, for example, it winds up that diffusion times get very long for distances over about 100 microns. And that sets effectively the separation between capillaries in the body. Every cell in your body needs to be within 2 or 3 diffusion lengths of the source of oxygen. So if I take a typical protein with a diffusion constant of 100 microns squared per second, and I ask the question, how long would it take for it to go from one end of a neuron to another? And it's a motor neuron. In your body, a motor neuron can have a length of a meter. So that means that a chemical would take, by diffusion, if you were going to send a signal in a neuron by diffusion down on a motor neuron, it would take 300 years for the signal to go from one end to the other. So that pretty well tells you that diffusion is not how we send signals over long distances or do transport over long distances. So diffusion is a fascinating process in the sense it's very simple. It doesn't take any energy. Diffusion doesn't use energy, which means it's very efficient. And it's always there. It's an inevitable consequence of the physics. And it's extremely effective over distances of 10 microns, even 50 microns. But over long distances, it doesn't work. Membranes are domains which have different local diffusion coefficients. In particular, membranes are obstacles to the flow of chemicals. That's why you have the membrane. It defines an inside and an outside, keeps things inside the cell. But the cell has a variety of ways of getting things across its membrane. It needs to get nutrients and oxygen in and some signals, like growth factors. It needs to get waste products out. Some things cross the membrane directly. They actually go through the lipid bilayer. There are ion channels. As I mentioned, those ion channels could literally be holes, like gap junctions. They can also be pumps of various kinds. And within the cell itself, there are a lot of large molecules that mean that you don't diffuse everywhere. You have to diffuse around the large molecules. So let's see how we're doing on time. We're almost done. So I guess we won't get to secretion and absorption today. We probably don't have time. Pedro, you started but didn't do the full exercise with membrane, right? You mentioned it but didn't do it. Okay. Would people like to do that membrane exercise as the last thing for today? Now that you've got that chemical, now you can draw the concentration. It becomes more interesting. So what we're going to do is we're going to create a permeable membrane. So what you want to do now is go back to your code that you've just written. And we're going to draw, use uniform initializer, create a single cell type called membrane, and use uniform initializer to draw a box. You can use single voxel cells to draw a box of cells down the middle of your simulation in the vertical direction. And plot the field horizontally the way you did now already. Okay? And then run the simulation and ask what happens. And you want to freeze the membrane. When you create this membrane cell type, make it a frozen type so it doesn't move. So let me show you afterwards what that should look like so you get a sense. Here, I'll walk you through the setup for it if you want. So we'll create a lattice, say 200 by 200 lattice, no flux boundary conditions. We'll use a rectangular slab, initial conditions. We'll create one cell type called membrane. Do people want to just, do you want me to start again and have people actually walk through it as you do it? I can stop, or let me walk through it first and then we can come back. So we create a single type called membrane, and we haven't used the freeze button, but we'll use the freeze button here to freeze the cell type. And we'll create a field here. I called it oxygen. You have a choice of that. And then in the uniform initializer, I'm going to adjust the box so that the box min is, say, 45. The box max is 50 or 55. Y goes from 0 to the top. And here I made the width of 7, but you could make the width 1 if you wanted. Doesn't matter for the size. Okay. And then in the diffusion constant, I make the global diffusion constant 1, the global decay constant 0. I set a constant value of 10 on the left, on min, 0 on the right, and I make the y-axis periodic. And I should get something that looks like that. In this case, the cells aren't doing anything at all. They're having no effect. So this is essentially the simulation you already wrote, putting the this line of cells back in.
The freeze if you're not using Blizzard again.
The command is /freeze. Let me get that for you.
Is it in the initializer or the cell type register?
It is in, let's see. If I go into Wizard, Emma asks, how do we freeze the cells? So let me,
I think you have to type freeze equals
Yeah.
quotes.
Okay, so in the cell type itself, let me see, I need to do a new cell type. So let me do this. CC3DML, plugins, cell type, replace, cell 1, or I call it wall, right? Wall, freeze, add. Okay. So freeze equals double quote. Or you can call it membrane, yes. And I'm sorry, the screen share, I have to redo the screen share for the people who are online. There was a question about how you create a frozen cell type. Cell type freeze equals quote, quote, and then the type name here. If you go into CC3DML, plugins, cell type, it will pop up the window that you need to add it so you don't have to remember, okay? Membrane, freeze, add, okay. Okay, did people see that who are online? That work?
Oh, no.
And then in this code we wrote, we got rid of the initial condition generator. So again, I can go to CC3DML. In this case, it's a steppable. I want uniform initializer.
There's one.
Oh, I see one.
And here I want box min and X can say 100. Box max 110. Y is 0. Box max 256. Let me make sure I made the size right. The size of the simulation is 256 squared, so I'll make it that. And then the width I could make 7. Okay. Does that work for you? I'll leave that on the screen for people to copy.
Oh,
Were people able to get that to work? Yes, no, Alex, no?
I
Not
think
work.
maybe.
You want to screen share it and show us what you got? We're going to be, we're yeah, that looks good. Okay. Okay, so let's do one last thing before we break. So now let's go into, Alex, why don't we do it for you? Why don't you screen share and let's do it in your code? So we can see everybody do it together. Okay, so now what we want to do is we want to make that, do you have your scientific plot still, or did you lose your scientific plot?
I'd have to get it from the other one.
Okay, that's okay. We don't need to do it for the moment. It'll be good to put the scientific plot back in, but we'll leave it for the moment. So what I want you to do now is we're going to use the fact that we can have the diffusion constant be different in different places. So let's go to where we define the diffusion field. And let us make the diffusion constant in the cell, the diffusion constant in the cell be 0. So where you have diffusion data, let's see how we have to do this.
I have to recall it, I think, to get the syntax.
So let's go to the CC3DML, hold down menu, go to steppables, and go to diffusion solver, and hit that one, yeah. And don't hit replace, hit comment out and add. Okay, now we're just going to look and see how to do this. You see where it says diffusion coefficient cell type membrane 0.1? Copy that line. Copy that line. No, the one above it.
where is it?
Copy that line. Just copy it. And now undo.
Here?
Control Z. And now, now in there, paste that line below global decay constant. Yeah, there. Okay, good.
No, it's not.
That's a trick. If you don't remember how to do something, you can paste the thing in temporarily, copy it, and then undo it. Okay, so now save it and now run that. Okay. Now what's going to happen when the chemical gets to the wall? Let's see what happens. Maybe nothing happens.
Yeah. Say,
Do you want to say?
no, we don't. Zero.
I think something happened. I think the chemical isn't getting through.
Oh,
Hmm.
there we go.
Yeah.
Oh, it's updating every.
Maybe it is. Let's see. Is it leaking?
Two.
Hmm.
Is my computer.
Looks like it might be leaking, huh?
The diffusion coefficient to 0.0, so that we can see the color.
Let's look back at our XML. Okay. Oh, because we made the coefficient 0.1, not 1. So it's there, so it's leaky. We made a leaky membrane. Make it 0.0. 0.0. It was doing the right thing, but I wasn't expecting that value. So you had a leaky membrane there. Now you have an obstructing membrane, so let's see what happens. Now you'll see the chemical won't get through at all. All right. So now the last thing I'd like to do, and then we'll stop, is let's make a little hole in that membrane. So let's go to the XML. And let's copy in the where you created your membrane at the bottom of your XML here. Good. Copy the whole thing from region to region. There we go. Paste it in again below. And now we're going to interrupt it. So instead of going from Y0 to 200, we'll go from Y0 to say 90. And from 95 to 200. So we're going to make a small hole. Now run it. Now you've got an ion channel. And what's going to be interesting here is look at the shape of the chemical field on the left-hand side of that hole.
It's nice to use the isolines.
Yeah, you could turn the ISO lines on in your configuration. Go to field, and where it says show contours, and maybe make ISO values, make it 20. There we go. Save. Did it not? Why did it not turn on my ISO line?
Go back to the configuration. I think that's not how it works. So you have to type ISO values and then 1, 2, 3, 4.
Oh, it's changed the.
I think it always has been like this. Go 1, 2, 3, 4 until 10.
No.
Did it update?
No, they're there. They're just hard to see. The color is the issue is the color. They're there. They're just not. They're just drawn. The issue is they're white.
Yeah, you can change the color of the isolines too, I think. No, you can't.
No, it's under. Yeah, contour color. Make them black.
Okay.
Yeah, there you go. Huh.
I think the contour color is the
All right.
contour of
Oh.
the box.
Let me play with that one. I wasn't expecting to turn the ISO lines on. The key thing you notice, it's a little bit hard to see here because of we made the hole a little bit small, is that you'll see that the concentration basically is circular on the left-hand side. So we have a uniform concentration. We have basically a flat interface coming to the left, from the right to the left. And then you'll see a high concentration just around the hole, and then a decreasing, essentially a circular concentration decreasing away from the hole.
I think it's possible to see it already. At least if you see in the right side, you can see that it looks like how do you say, a sunrise, or the opposite one?
No.
It's kind of circular.
Okay. So this is basically and we'll stop for today here. This is a simulation that you could actually do a lot with. You can make the size of these holes bigger or smaller. you can have leakage in the membrane, which you would normally have. And now if you add that plot where you plot the concentration as a function of position along the x-axis, you can see how that concentration evolves in time for different geometries. And this can begin. What we don't have the ability to do in this simulation is to have pumping. In other words, here we have passive diffusion. We have chemical that is moving through diffusion, either in the membrane or in the hole. What we don't have the ability to do is have what we call jump boundary conditions, where we have active transport, where we have a pump that's actually moving the chemical across the boundary. We'll be able to do that later in the class, but at the moment we don't have the ability to do that. Okay, well, thank people. I realize that we had a few snafus again today, but I hope that people learned a little bit. I'd encourage you to try playing with these kinds of simulations and becoming familiar with it. Really, uptake and secretion are not very different from controlling boundary conditions. And so if you master the ability to control boundary conditions and to plot, I think next time we'll be able to do the secretion plug-in pretty quickly. Any questions before we break? For next week, I would like to ask people to be ready to,
This is on.
would like people to be ready to be able to tell me a little bit about their project ideas. A couple of people have talked to me about projects, but not everybody. And so if you're enrolled in the class and you're doing a project, it's very important that by next Monday you talk to me about project ideas. So please do that. Okay, thank you, everybody. Sorry, Kyle, go ahead.
It may be like in the bonus overtime. I'm curious about semi-permeable barrier membranes and how you model things that may have like certain things get through and certain things don't. And then I'm curious also about how very large molecules, how you model their tendency to be more fragile. When you're dealing with the smaller compounds or chemistry, things are pretty consistent. But when you get like these big organic things that are like in the lungs, the big compounds kind of break up sometimes.
So Kyle asks, what do we do about semi-permeable membranes? Well, in this case, we can define the membrane diffusivity to be different for different chemical species. What we can't do is do something like a symport or an antiport, where the movement of the chemical is not a simple linear function of the concentration difference across the membrane. In other words, if the flow of the chemical across the membrane depended on the concentration of a different species, that we can't do with this. Now we could do it using the reaction diffusion solver. But that's one I should probably have an exercise for. I don't have an exercise to illustrate that. That would be a good one to write.
You could to build a pump, have two cells side by side.
Right.
One uptaking the water, the other secreting the.
Right, but I'd have to pass information. I'd have to by hand pass information between the cells about what was happening.
Yes.
The other one that I could do that Kyle asked about was, what if chemicals are unstable? And we have so far turned off the decay coefficient, but we can set a decay coefficient for the chemicals. And we can also have active decay. In other words, we can say that the decay coefficient, for example, we could have a subregion of a cell that's defined as a lysosome, and say that the decay coefficient is high in the lysosome region and low elsewhere. And again, we could also define, using the reaction diffusion equation, that the breakdown of the chemical was dependent on the concentration of a second molecule. So we can implement that. What we can't do is have an explicit representation of the spatial structure of the second molecule. So we have to assume that the scale of our voxels is big enough that we have plenty of molecules within those voxels. Now, in reality, sometimes there really is only one molecule, one of a molecule in the cell, and then talking about its concentration doesn't make a lot of sense. But that's a great question, Kyle. And
Well,
again, there's a lot one could do with these, even these apparently very simple simulations. You could actually explore a lot of important biology and biochemistry.
I was reading a bunch of papers a year or two ago about senescence in cells and how if they get too much garbage that doesn't get moved out, or the neighborhood has a lot of like vacant cars in it and dumpster fires, it tends to accelerate the aging and the death of the cells. And so I was just thinking about how would you understand the dynamics of that, you know?
Right. So Kyle points out that one theory of senescence is that you have accumulation of damaged and incompletely degraded proteins.
He turns it into the right language.
And that actually occurs in multiple cases. In the eye, the photoreceptors have very high metabolic rate. And the waste products of the photoreceptors have to be eliminated through the vasculature. And that means that the waste products have to cross Brooks membrane at the base of the retina. As that happens, a bunch of essentially partially degraded molecules accumulate on Brooks membrane, which reduces the adhesivity of the pigmented retinal epithelial cells to the Brooks membrane, which is why elderly people are much more at risk of a detached retina than younger people. Basically, because the cells at the back of the retina have to stick to Brooks membrane through a bunch of gunk. And if you want to glue something to a surface, you don't put
Clean.
you want it to be clean. Yeah.
That's cool.
Another place that happens is in bacteria.
Yeah.
When bacteria grow, they do the synthesis on one end of the bacterium.
So
And so the old proteins are on one side of the bacteria, but the new proteins are on the other side. And if you look at the probability of death of bacteria, the old end of the bacteria die more frequently than the new end of the bacteria. And so you can actually see senescence even in individual bacteria.